Telomeres, structure and function. So telomeres are that part of the chromosome that actually stabilizes the chromosome and protects the chromosomes. So let us look at the learning outcomes of this session. The telomeres are ends of chromosomes that are non-coding repetitive DNA which protect the functional genes of the chromosomes from damage. They prevent unnecessary homologous recombination or any kind of recombination. Even end joining recombination is prevented. Unnecessary DNA repair at the ends is being prevented, and also fusion of the ends of two different chromosomes are prevented. So interchromosomal fusion is also prevented. As a cell divides with every replication cycle, the telomere shortens. This is because of a typical problem that is associated with termination of replication in eukaryotic DNA because they are linear DNA. So it has been observed that the ends of the lagging strand are never filled up, and hence one of the strands of that daughter DNA becomes shorter. So with every cycle, the end will become more and more short. When the telomeric length keeps decreasing and it reaches a critical limit, the protection due to these telomeres decreases, and that leads to cell senescence, or what we also understand by aging, and apoptosis. So this is something that is extremely important, and this has been taken up by various scientists, including Dr. Blackburn, who won the Nobel Prize for having understood how the telomerase functions in ensuring to maintain the length of the telomere. Telomerases are responsible for synthesis of the telomeres, and the telomerases are found to be active in germ cells and stem cells, and also have been reported to get activated in cancer cells and in vitro growing established cell lines. So we know that established cell lines gain the characteristic of becoming immortal, and somehow this immortality is associated with the presence of telomerase in many of these established cell lines. The telomeric DNA is associated with several telomere binding proteins that make the telomeres more protected from exonucleolytic degradation or mutations. So the first time that the telomeres were kind of discovered was by a scientist called Hermann Muller, who was experimenting with X-ray based mutagenesis, and he found that the ends of the chromosomes have unique properties, and he named these ends as telomeres. As in Greek, the word telo means the end and mere means part. So part of the end of the chromosome, okay, that's how the name comes, and that's how we today also call it as telomeric regions. So it is basically because of where it is positioned that it is called as telomeres. And when he was basically looking at how the mutagenic X-rays affect the chromosomes, and it was looking at how there is double strand breaks that is happening because of X-rays, he found that the ends of the chromosomes one would not observe mutations and that they were resistant to mutations, and that is why he felt that those ends of the chromosomes had this unique property of resisting any kind of mutation. Now, when you look at the telomeres per se, you will find that the telomeres are repeats of, or they are sequences that are getting repeated. So this portion is what is the telomeric DNA, and just preceding the telomeric DNA is the region which is called as subtelomeric DNA, and preceding the subtelomeric DNA is of course the chromosomal DNA, which would have all functional genes. So even if we had an exonuclease cutting onto the telomeric region, it will take time before it reaches the chromosomal DNA. However, if the telomere becomes shorter and shorter, which we have understood with every replication in eukaryotes, the telomere in a normal somatic cell will get shorter and shorter, then what can happen is that the exonucleases can affect the chromosomal DNA as well. And in such a case where the functional genes are mutated or functional genes are removed, then cells will lose certain functions, and because the cells lose functions, they will start aging. This TTAGGG is the repeat sequence that one observes all along the telomere. This is a conserved repeat sequence. Okay, all telomeres you will find the same sequence. Of course, albeit you may have certain changes in the sequence in different species, but more or less the sequence is almost the same, and that's why that is called as the conserved repeat sequence. You can have these repeats anywhere from 20 to 70 tandem repeats. Tandem repeats is because there is no other sequence between the adjacent repeat sequences. Therefore, they are called as tandem repeats. And the telomere always has one DNA strand which is longer than the other. So therefore, you have what is called as an overhang present. And this is generally the 3' end and which has the overhang. Since it is a GGTTAGG, so because you have a lot of G present in the single strand itself, it is called as a G tail or it is called as a G overhang, and the length of the overhang can vary from species to species. It can be anywhere between 30 to 300 nucleotides, and not just species to species, actually chromosome to chromosome also it can vary. But this is one interesting fact, and another interesting fact that has been observed is that in certain kind of organisms, you can also have the C-rich overhang present. So there you can have a C tail or a C overhang as well. So you can see how the complementary DNA that is formed against the telomeric region will be more rich in C. Okay. And so this portion can be called as the C-rich region, and this portion is what is the GT-rich region. Now, these tandem repeats are able to interact with several different telomere binding proteins. So, because they are associated with telomeric binding proteins, this region becomes more protected. And that is what one wants, because one doesn't want that you have enzymes or proteins coming and attacking or coming and biting into the functional DNA parts. Also, what is important and interesting to note is that this tandem repeats help in formation something called the T-loop, which is again a higher order structure. So we will look at it in a while. So when you look at the ends of the chromosomes, one will find that every telomeric end has this specific looping of the telomeric overhang, okay, and that is what is called as the T loop. So this is a very special structure that is formed at the ends of every chromosome. There have been microscopic evidences of the fact that the T loops are formed. So Jack Griffith in 1999 was able to actually show that you have the telomeric ends having what is called as the T loop. And the size of the T loop can vary depending on the length of the telomeric region, and we already know that the telomeric region can vary in its length itself. Now, this T loop is associated with a complex made up of several proteins, and that complex is called as Shelterin complex. So, let us look at how the T loop is formed and also what is the Shelterin complex. The Shelterin complex is made up of 6 subunits, different protein subunits. So you have TRF1, TRF2, POT1, TPP1, TIN2, and RAP1 that comprise the Shelterin complex. Now, interestingly, what you can observe is that the telomeric region only at the ends has the overhangs, otherwise it is a double-stranded DNA. So, the telomeric repeat factor binding protein TRF1 and TRF2 specifically come and bind to the double-stranded telomeric repeats. So, it does not bind, or TRF1 and TRF2 cannot bind to the single-stranded overhang, but it only binds to the double-strand region of the telomere. Now, this is important. Why is it important? Because the The TRF1 and TRF2 is able to distinguish a telomere from other parts of the chromosome because it can bind specifically to the tandem repeats. That is, it recognizes the TTAGGG sequence, and that is why it can come and bind to it, but only when it is in the double-stranded form. That is one aspect that is very clear. And secondly, what is important to note is that the ends of the telomere, which has an overhang, okay, is associated with, or rather, POT1 is the sheltering protein that comes and binds through only the single-stranded DNA. That is a single-stranded G overhang. The the TRF1 and the 2 recruit all the other sheltering proteins like TIN2, TPP1, RAP1, etc. Now, the overhang which is present, okay, that has a sequence that is complementary to the other DNA. So, in turn, what we are trying to say is that if this strand has TTAGGG, the other strand will have AAU, AATCCC. What does this mean? It means that the overhang which has TTAGGG, when the entire telomere loops, the overhang which is single strand can invade the upstream double-stranded telomeric region, and on invading it can displace the GT-rich sequence or the GT-rich strand but bind to the CA-rich strand. You see, in a telomeric double strand, one strand will have the GT-rich repeats and the other strand, because it is complementary, will have the CA-rich repeats. So, the overhang which is GT-rich can complementarily bind to the CA-rich strand. So, therefore, it can invade a double-stranded telomeric region, displace the GT-rich strand, and in turn bind complementarily to the CA-rich strand. So therefore, what will happen at this point is that the overhang, the end of the telomere, is now no longer free. Instead, it is forming a complementary binding with the telomeric region itself. So you have this complex that is formed over here. So since it is displaced, the GT-rich sequence of the double-stranded telomeric region is displaced. It is called as a T-loop. Immediately, therefore, this GT-rich sequence that has been displaced is single-stranded. So POT1 will come and bind to this single-stranded region, stabilizing it. And to that, you have the POT1 now binding to TPP1 as well. And the free overhang is now interacting. It is not free, it is interacting with the telomeric region itself, thereby forming what is called as the T-loop. So obviously, two things are very clear. One is that because of the formation of the T-loop, now you do not have a free end or a free overhang. The overhang itself is binding with the telomeric region itself to form the loop. So it is more protected, correct? You don't have a free end that can be attacked by any exonuclease. So you cannot have any exonucleolytic degradation because now there is no free end. It is present within the telomere itself in the form of a loop. Interesting to note, therefore, this portion has 3 DNA strands at one point. So you have here an entire complex which has 3 strands at this point itself. The rest of the telomere, that is the double-stranded region, also is bound to by the different proteins of the Shelterin complex. So the entire telomeric region, you must understand, is bound to by the proteins of the Shelterin complex. So you have these telomeric ends being protected by the Shelterin complex, okay? And also interesting to notice that many of these sheltering proteins have regulatory roles to play. That means that these proteins ensure that it is preventing the DNA repair molecules to come and bind to this region, because see, if you have an overhang is generally considered to be a cut end, and if it is a cut end, then that is considered to be a DNA damage. If it's a DNA damage, you can have homologous recombination happening using that overhang, or you can have DNA repair systems coming and binding to it, cutting it out so that you have that portion removed, right? So, or you can have other signaling pathways that come and kind of signal the cell to do something about this region. Whereas if you have the Shelterin complex form, the Shelterin complex tries to avoid having all these molecular mechanisms coming and interacting with the telomeric region, thereby protecting the entire chromosome at the ends. So you can very clearly observe that the G tail can be bound to by the POT1. If it is not forming a loop, then the ends are protected by the POT1. But if it is forming a loop, again, the D loop, that is the displaced loop, is bound by the POT1 and stabilized. Because we know single-stranded DNA are more prone to exonuclease attack or are more prone to nuclease attack. So when you have a when you have a protein associated with a single strand, then the single strand is also protected and the entire complex is stabilized. So this is how a T loop is formed at the ends of the chromosomes. Now there is one other, many studies have have shown that other than just having the overhangs, you can also have G quadruplexes. So we all know that G quadruplexes are higher order structure, especially formed by single-stranded DNA and single-stranded RNA. So you can see how the single-stranded DNA, the overhang, can be present as a quadruplex. And when present as a quadruplex, the single strand is kind of associated with each other. So again, it is a structure that cannot be easily contacted or easily, cleaved by the exonucleolytic or cannot be cleaved by the nucleases. So you do not have exonucleolytic degradation happening because of the presence of the G quadruplexes. Many studies have shown that the G quadruplex itself can also engage in formation of the T loop per se. You have more of the displaced DNA because of the kind of structure that the quadruplex forms, but this structure is also stabilized by the Shelterin complex per se. So it is very interesting to note that the G overhangs are formed only under certain conditions, of course, and they perform as a capping role. So they have a capping role to play. And the G quadruplex, along with the POT1, is reportedly involved in telomere lengthening. So together, the G quadruplex and the POT1 can recruit telomerase and initiate lengthening of the telomere. Okay, so that is something important. So you must also understand that Shelterin complexes themselves are able to regulate the lengthening of the telomere by either recruiting telomerase or not recruiting telomerase. As mentioned earlier, the telomere length is maintained by telomerase, and telomerases are not active in all cell types. So telomerase is constitutively active. When one says constitutively active, it means that this gene is getting expressed all the time in the germline cells. So you will always find presence of telomerase, and you will always therefore find that you have telomeric lengthening every time replication happens. So in such cells, you will never find that the telomere is shortening because telomerase is always available to increase the length of the telomere or to synthesize the telomere per se. But when you talk about normal somatic cells, in normal somatic cells, the gene expression of telomerase does not happen, it is repressed, and so you will not find active telomerase in normal somatic cells. So when these somatic cells undergo cell division, okay, at the end of every replication, what you will find is that the telomeric length is going to shorten because end replication does not happen. And since end replication does not happen, every replication cycle, you will find that the telomere length is decreasing. When the telomere length reaches a certain critical length, then one observes that the cell starts to age or the cell undergoes cell death. So this is something that is very clearly associated with telomere shortening. Okay, before telomerase is reactivated in many cancer cells. So you will see that as the cells keep multiplying in cancer, we know that it is an an uncontrolled, not regulated cell division. So with every cell division, you should actually have the telomere length decreasing. If the telomere length decreases, then the cancer cell should die. But somehow the cancer cells, many of the cancer cells reach immortality because you have the telomerase again active in those cells, and because they are active, the telomere length doesn't shorten, and because they do not shorten, the cancer cells get the immortal condition, so they live, they do not age. Okay, so that is something that has been observed with many cancer cells. Now let us look at how telomerase functions. So we all know that the telomeric ends have a G overhang. Now, interestingly, the telomerase comes and binds to the G overhang. In fact, they are recruited by, TPP1 of the Shelterin, POT1 of the Shelterin. These molecules are actually able to interact with the, G overhang, or they are recruited to the telomeric end, and they bind to the G overhang because within the telomerase is present an RNA. So actually telomerase is not a pure protein, it is a ribonucleoprotein because one of its subunits has an RNA. So you have what is called as RNA or ribonucleoprotein core in the telomerase per se, and the inherent RNA that is present has a sequence, okay, which is similar to the complementary sequence of the telomere. So if this is TTAGG, what you have as the complementary sequence is AATCCC. Look at the fact, AAUCCC. So this is same as this. So which means that this can now complementarily bind to the GGG. Correct? Once it binds to the GGG, okay, so you have this telomere encoding RNA. So therefore, this is what is going to act as a template. You can understand that if a DNA polymerase has to synthesize anything further, you need a template. Now, in this portion of the overhang, okay, you have this as a template, okay. So you should be able to synthesize, but this is the 3' end. You see, this is the 3' end. So you can only extend it over here. This is the 5' end. So from 5' end, we know that DNA polymerases cannot synthesize. So if it has to synthesize, it should have a 3' end strand over here to synthesize in this direction. So I guess you understand what the end replication problem is. Because you have this as the template and this itself is 3' or a 3' end, you don't have a 3' end over here. This is 5'. This portion cannot be synthesized by a normal DNA polymerase. But the telomerase having the RNA which has the sequence complementary to the telomeric sequence, okay, it comes and binds complementarily to. In fact, therefore, you must understand that it is the telomeric region, encoding region, which gives rise to the telomeric repeats. So the CCC comes and binds to the GGG, okay. The telomerase has a subunit which is acting as a reverse transcriptase. So this region, this subunit is called as the telomerase reverse transcriptase, TERT, and it has been observed that one of the domains, the reverse transcription domains, has the typical palm and finger domains as in the DNA polymerase 1. Okay, or the DNA polymerases have a typical palm-like structure, so that is observed even with the telomerase. Now, therefore, this reverse transcriptase equips the telomerase to actually use the RNA as a template and synthesize the DNA. So, considering that that is the case, you would have the new DNA, telomere DNA being formed, okay, complementary to this. So, once this has been formed, you will find that the telomerase displaces and rejoins again. So, you can see it has increased in length. Now, it will come and bind to this, right, and extend the DNA further. So, that is how it is going to continuously extend this strand. And when you have this strand getting extended, you can have a primer coming and forming here, And using the 3' OH end of the primer, the DNA polymerase can extend this. So you will have more and more double-stranded telomere being formed. But you will always at the end have an overhang as the telomerase keeps on extending the telomere. The telomerase structure, as mentioned to you, it has the TERT. This is the reverse transcriptase, and this is the TER, that is the RNA that acts as the template. And this telomerase is actually recruited by TPP1, POT1, and also the RAP1 of the Shelterin complex. So they have a role to play in recruiting the telomerase. And once the telomerase is recruited, look at the fact that it has a kind of a palm structure. So this forms the fingers, this forms the thumb, and this is the palm. So it has this typical palm structure that is same as the DNA polymerase structure, especially the DNA polymerase 1 structure. So telomerase has been associated with aging, as already explained. As the telomere shortens, you would have the cell senescing and aging, and also undergoing apoptosis if needed. So you can have cell death happening. Telomerase is associated with cancer, as mentioned, and certain lifestyle disorders or factors such as smoking, obesity, lack of exercise, and consumption of unhealthy diet can increase the pace of telomere shortening. So, this is something that has been observed in many studies. So, a person who is smoking may live 7 to 8 years lesser than if he were not smoking. So, because it has been found that those smoking would have a rapid shortening of the telomere happening. So let's make the conclusions. Telomeres are made of tandem repeats of GT-rich sequence with a G overhang that extends by 30 to 300 nucleotides or 20 to 770 tandem repeats. In eukaryotes, the G overhangs can hybridize with upstream tandem repeats to displace the double-stranded DNA to form a D loop. And overall, the entire telomere folds back to form a T loop. The T-loop is bound to proteins of the Shelterin complex that plays an important role in protecting the telomeric DNA as well as regulating the telomere synthesis. Telomerase is a specialized DNA polymerase. It is a reverse transcriptase that has the RNA template within its structure to synthesize the DNA. That is a telomeric DNA. Thus, telomeres found at the ends of the chromosome not just stabilize chromosomes by protecting the functional genes of the chromosomes, but what has been interesting fact is that shortening of telomeres is associated with aging and certain diseases, including CHD, coronary heart diseases. So these are certain aspects of telomeres which make it a very interesting high order structure in DNA. Thank you.